Okay, so enzymes are important for life. The basic thing, the enzyme is able to convert sucrose into glucose, and the enzyme is sucrase, or normally we call it invertase enzyme, which will convert sucrose into the glucose and fructose. Okay, so welcome back to the class. Sorry, something went wrong and I'm starting it again. Okay, so everything I have discussed, do it again. Then, oh sorry. So this oxidation is done, the oxidation of sucrose or other sugar to get the glucose finally, and glucose is the basic requirement of our body to generate ATP. So in this chapter, we turn our attention to the reaction catalyzed biological systems. These are called enzymes, which are responsible for catalyzing the biochemical reactions in biochemical systems, for example, in animals. The most remarkable and highly specialized proteins, because the most of the enzymes are proteins, so they are highly specialized and remarkable proteins. Enzymes have extraordinary catalytic power, as we will discuss some of the enzymes. So if you want to know how, who is the maximum, which enzyme is having the maximum catalytic power, so this you can get from this particular PDF. Okay, so this Pathfinder is, it is given somewhere, if I'm not wrong. So we will discuss these things: acid-base catalysis, covalent catalysis, and so on. So sorry, actually not covered in this particular PDF, but actually the catalase enzyme is having the maximum catalytic power, which can convert the maximum number of substrate molecules per second. Okay. So this is only written like they are having high degree of specificity, which we have discussed. They accelerate the chemical reactions tremendously, maximal for catalase actually, and they function in aqueous solution under very mild conditions of temperature and pH. For example, which is present inside our cells, that is around 7 or neutral pH, or 7.2 exactly, and the temperature is around body temperature. Then we will discuss about enzyme kinetics, okay, and something we will cover about the mechanisms and the inhibition types, as well as, okay, so because we are going to cover the inhibition part also, so let me show you one important table which will help you to understand the type of inhibitions which we will cover in this particular chapter. So it is given here. Okay, so generally the inhibition can be divided into two types. One is competitive, and second is, okay, so non-competitive or uncompetitive. So the competitive inhibitor, and also there is a new category that is called mixed inhibition. So direct question has been asked from this particular table actually. So you should know certain things when we are talking about the inhibition part in enzymes. So for example, okay, so we are discussing this. Okay, so first thing is the type of inhibition. It may be competitive, non-competitive, uncompetitive, or mixed type, which is a combination of these two, non-competitive and uncompetitive. So competitive is something in which the V maximum will decrease. I will tell you what is V maximum, what is Km. So whenever we talk in terms of enzyme inhibition, so you should know this particular diagram. So this is called Michaelis-Menten equation or the curve, in which the enzymes which are following this equation. So if you draw the velocity versus the substrate concentration, so the substrate concentration at which the velocity is half Vmax. Okay, so this is half Vmax. And this substrate concentration, if you draw some perpendicular line from here, you will get a concentration that is called the substrate concentration at which you will get the half maximum velocity of that particular reaction. So this is actually called the Michaelis-Menten constant, Km value. So from the Km value and from this Vmax value, so this Vmax is actually the upper curve of this approachable point of this particular curve. So this is called Vmax. And from the Km, you can draw some type of equation or some type of relation, which are shown in this particular table actually. So you only need to remember the two things. One is Km. Km is that particular substrate concentration at which the maximum velocity is just half Vmax. Okay, so if this is a type of competitive inhibition, you will find that this Km, which is the substrate concentration at which the velocity is half V maximum, it will increase, and the Vmax value will decrease. If this is increasing, this is decreasing. This type of inhibition is competitive inhibition. So based on these differences, you can categorize these type of inhibitors into four categories. So the second is non-competitive inhibition, in which the Km will not change. The concentration of the substrate will remain unchanged, at which you will find the Vmax, but there is a decrease, the decrease in the V maximum value. Okay, so the Vmax will decrease, but it will remain unchanged, as you can also find from this particular diagram. Okay, so you can see here. So if the V maximum is unchanged, sorry, this one. Okay, so this is competitive, this is non-competitive, and this is uncompetitive. So majority of the inhibitions are three types, and one which we have added is mixed, which is combination of these two. I will discuss that also. So in competitive inhibition, you can see this. Okay, if there is a competitive inhibitor, so it will decrease the Vmax. So this, sorry, this will not change. This curve only shift here, but it will approach this V maximum. It will join this particular curve, which is present in the absence of inhibitor. So the V maximum will remain the same. There is no change in V maximum. So directly in, for example, in the question, these graphs may be given directly, and in the question you may be asked which type of inhibition it is. So from the graph, actually, you need to recognize what type of inhibition he's talking about. And if you know these things, if you know about that particular table which we were discussing actually, so you can find it out whether it is competitive, non-competitive, or uncompetitive. So if this line will remain the same, actually this is dashed line is showing the inhibitor, okay, in the presence of inhibitor, that is the competitive inhibitor actually. So it will join this V maximum, okay, so this line will join this. So there is no change in V maximum value, it is remain unchanged, but you will see this Km value will shift here. So because the V maximum is here, so if the we find something half value here, we draw a perpendicular line, we draw this line, and then we draw this line, then we will see this Km and Km dash are different. So the Km is increased. So this is competitive inhibition. In second type, that is called non-competitive, in this value, in this actually this graph, which is a dashed one in presence of inhibitor, it will be shifted on the right hand side in a similar fashion in this way, but it will not join this V maximum. So the V max will decrease, whereas you will find the first case in this purple line, actually this is V max. And if you find a line or the some half value, half maximum of half Vmax, it is here, and the Km value is here. Okay. Again, if this is decreased in presence of inhibitor, now the Vmax dash, which is a decreased value, is shifted to this line. Now, if you find the half value of this will come somewhere here below this line. And if you join this, you will find that the Km will be on this axis, on this x-axis, will be equal to the Km dash. So it is not changing. Km will remain unchanged or is the same, whereas the Vmax value is decreased. So this is non-competitive. In NC or non-competitive, the Km will remain same, whereas the Vmaximum will decrease. If you Okay, so again, then there is another kind of inhibition which is very unusual. This is uncompetitive inhibition. So in this case, this curve has a shift like this. So in this case, this Km value is decreased. Okay. Also, the Vmax value is decreased. So if this will decrease and this will also decrease, it will move towards the left-hand side. Okay, so this curve is something like this. When you find the half maximum of this line, it is here. So this is the Km of normal enzyme. But in presence of inhibitor, the value of this will show this half Vmax. And if you draw a perpendicular line here, you will find that the Km value is less than the first case. So from these three cases, you are able to understand what is happening actually, how these graphs are changing. Also, these Addy-Hofstee plots we will discuss in the kinetics portion when we will discuss the Michaelis-Menten equation. But because this is the first class and I'm trying to make a base so that you can get some of the concepts what we are going to discuss in our upcoming classes. So here, if you see in case of competitive inhibitor, if you draw this curve in this form, it will show you these two lines. So it is very easy to find actually. So with the help of these these plots, so if they are intersecting each other, two lines are intersecting at this particular line. So this is called competitive inhibition. If the two lines are emerging from the same line and forming something, a V shapes like structure is formed, then this kind of inhibition is non-competitive. And if both the lines are running parallel, then this kind of inhibition is uncompetitive, because in this case, both are decreasing. Both the V maximum and the Km values are decreasing. So you need to remember all these 6 diagrams. You should keep in mind, and you should get this particular concept of remembering this particular diagram from this table. So you need to remember this table, and if you remember this, you will not be confused. Okay, so everything is clearly written here, and you can draw something to better understand. Okay, so you can write here, this is competitive inhibition, that is the first type. This is C, this is NC, this is uncompetitive inhibition. So uncompetitive UC. Okay, and this is mixed. So even you can remove the C from these. So this is C, this is NUM. In this case, if you remember NE1, either V maximum or Km value, okay, so you will get the concept. In this case, it is increasing, and competitive, so it will decrease. So both values are opposite. In the second case, that is non-competitive, it is decreasing like similar, it is common to this, it will decrease, but it will remain unchanged. So there is no change in this particular value, it will remain the same. Km is equal to Km dash. In this case, both are decreasing. So this is decreasing, decreasing, decreasing in all the cases. So this will decrease. And in this case, this will also decrease because both are decreasing. Okay, in mixed inhibition, actually, this is, so this is common to all. This is decreasing. There is no change in V maximum. V maximum is decreasing in all the cases. So overall, this is decreasing. Whereas you need to remember the Km values. In the first case, it is increasing. In the second case, there is no change. In the third case, it will decrease again. In the fourth case, in mixed inhibition, actually, this should generally increase. This should generally increase, but actually, in some cases, only in exceptions, actually, it will decrease. So actually, you need to remember it is increasing in this particular case. Okay, so because there is a decrease already in uncompetitive inhibition, so last one actually it is an increase, it is shown by an increase. Only few rare cases, so you need to remember this one only. Okay, second one you can avoid. So if you remember this particular table, you can get the particular concept of inhibition also. Then we will moving again to our. Okay, fine. So there is a query. Okay, let me. This is different from the table diagram. The table it is mentioned V maximum decreases and here unchanged. Competitive. Okay, so I actually not. Is it so? Oh yes, actually it is written like it is decreasing, but yes, Trishla, you got the right point actually. So, so there is something wrong in this table. Let me correct this. Actually, I did not recognize. I just blindly believing like it is correct. Okay, so in this competitive inhibition, actually this is not decreasing, this is unchanged. So there you can write like 0, there is no change, and it is increasing. Okay, now we are drawing it correctly. Then in non-competitive inhibition, it is decreasing and there is no change in this particular Km, that is fine. And okay, so okay, now it is easy to remember also. In this case, this is unchanged. In this case, this is unchanged. Whereas in competitive inhibition, this is increasing. In the non-competitive inhibition, this is decreasing. Now it's fine. Then in the next one, both are decreasing. This is good. This is fine. And the last one, I just told you that in this particular case, because this is decreasing, so this is decreasing in this case, and this also. Whereas in this case, it is increasing. Mostly it is increasing. That's fine. So you can remember this one also. No, now it is fine. Okay, actually, I have checked from my notes also. So I'm confirming this is fine. Okay, thank you, Trishla, to remind me. Okay, fine. Now it's fine. So if you remember this, you will not forget anything about inhibition, and the right question generally have asked from this particular section. So because this is our first class, so I discussed this also. Now let me move from this particular portion. This is not our topic of interest in this particular class actually. Okay, so now the question is how this enzyme term is given and what it mean, and what type of enzymes were the first enzymes which were studied, and we are able to got these concepts which we are discussing in today's class. So much of the history of biochemistry, actually biochemistry, and most of the biochemists are initially in the 70s or early 80s. They were crystallizing different type of proteins. They were trying to identify their functions and so on. So most of the history of biochemistry is the history of enzyme research. So they were discovering these enzymes actually. And I have heard this also that earlier in up to in 1980s, So if you are able to recognize a particular protein, if you are able to characterize and crystallize a particular or specific protein or enzyme, you will be able to got your PhD degree. Actually, this was like that only. Okay, so but now it is becoming uh, difficult as more and more research and different type of things are happening in the field of science. Okay, so biological catalysis was first recognized and described in 70s, where actually they were trying to discover the digestion of meat by secretions of the stomach. So they are trying to recognize that this and the stomach is secreting something which will help the meat to digest. Then research continued, and for a century, and in 80s, uh, with examinations of the conversion of the starch to the sugar, that is the glucose and fructose, by saliva and various plant extracts, they were able to recognize the enzyme invertase or sucrase. Then in 1850s, one scientist called Louis Pasteur concluded that fermentation of sugar into the alcohol by the yeast is catalyzed by the fermenters. Okay, so actually he gave this particular definition, but he was not knowing actually the specific enzymes which are responsible for the conversion of this sugar into the alcohol. Today only we know this is the alcohol dehydrogenase enzyme, which is specifically converting the sugars into the alcohol. Okay, so he postulated that these fermenters or ferments were inseparable from the structure of living yeast cell, which is not correct in today's scenario. We already know this. Okay, so he gave this particular definition because at that time in 1850s, when Louis Pasteur was trying to finding how this sugar is getting converted into the alcohol, so at that time there is a concept called vitalism, and according to the vitalism theory, it was believed that there are certain forces, some extraordinary power in living beings. So only in the living beings these things, for example, urea formation or these things are happening within living organisms only. But only this view is called vitalism. It is written here. This is vitalism, and this prevailed for decades. So for decades, actually, scientists are believing like these reactions or these chemical reactions are only can happen within living organisms. So these cannot happen outside or in the test tube. So this particular concept is called vitalism. Then there is one scientist which is very prominent, and actually the the figure is shown. So this is Edward Buchner. So he survived from 1860 to 1917. So he was only able to discover that cell-free yeast extract could ferment the sugar to alcohol. तो उन्होंने पहली बार बताया कि जो अगर हम यीस्ट के एक्सट्रैक्ट को किसी टेस्ट ट्यूब में शुगर से या सुक्रोज से रिएक्ट करवाएं तब भी वो अल्कोहल बना सकता है. So this kind of concept, which is called vitalism, is got a serious blow, and then after some years also this vitalism prevailed, but at the last the scientific community is able to believe that there is nothing unusual or extraordinary in living organisms. These type of reactions can be done in the test tubes or in labs also outside the living organisms. So in 1897 it was the actual start of enzymology. Actually, scientists start identifying different type of enzymes, and it is all with the help of this particular scientist, this Edward Buchner, who discovered that these reactions or the yeast extract can ferment the sugar into the alcohol in outside the living organisms also or in the test tube, providing that fermentation was promoted by molecules that continue to function when removed from the cell. So first type of this is a different type of concept at that time in 1897 when he was talking about some of the enzymes, and actually he was not knowing what kind of molecules are these were. So today we know these are called enzymes, and this specific enzyme which is responsible for sugar fermentation into the alcohol, for alcohol generation, this is alcohol dehydrogenase. So let me show you the reaction, how alcohol dehydrogenase is able to work. So this reaction actually, so are you able to see this particular screen? If not, I'm zooming it out. Zooming in. So the glucose will, first of all, the glucose will form from the sucrose, and with the help of enzyme, this is called either maltose or the sucrase enzyme or invertase. Finally, the glucose will produce from maltose or sucrose. And this glucose will be converted with the help of this glycolysis pathway, will be converted into pyruvate, where you will get some ATP. Then finally, this pyruvate will be converted into acetaldehyde after release of the molecule of CO2. The CO2 will be released in this particular process. Then finally, in the third step, the acetaldehyde will be converted into ethanol. For easy these diagrams are shown here. So these molecules, so first of all, this glucose, then pyruvate, then acetaldehyde, and finally the ethanol will be formed. So these are shown in this particular diagram. And the enzyme which is responsible for this conversion is called as, so it is written here somewhere, no? So actually it is alcohol dehydrogenase, which is using NADH as reducing power, and it will finally convert the pyruvate into the ethanol. Okay. But these scientists, or the scientist at that time, for example, Edward Buchner, he knows nothing about these particular enzymes or molecules which are converting the glucose into alcohol. Then, and he told for the first time that when these molecules or these type of alcohol dehydrogenase like enzymes are removed from the cell, they will continue to work if they are not denatured, or if there is no change in their structure. Okay, so Buchner experiment marked the end of vitalism or vitalistic notions, and dawn of the science of the biochemistry, and since then the science of biochemistry has been started. Then there is another scientist that is called Kuhne, that is Kuhne actually, Kuhne, Frederick W. Kuhne, who gave this particular term enzyme for that particular molecules which were detected by Buchner. So Buchner only identified certain molecules, but he know nothing about mechanism, what is happening. Then another scientist came and he gave the term enzyme. So in some of the exams, you may find this direct question, name the scientist who gave this particular term. So this term was given by this Kuhne, and from the Greek enzymos, which is having some meaning. Okay. Then there is another scientist after the Buchner, James Sumner. So this James Sumner and JBS Haldane, first of all, James Sumner have isolated and crystallized the first enzyme. So if someone asks you, name the first enzyme which was crystallized, so it was the James Sumner who isolated and crystallized the enzyme urease. So urease was the first enzyme to be crystallized in 1926. Okay, so it was a breakthrough in early enzyme study. No one has crystallized the enzyme earlier. So Sumner found that urease crystals consist entirely of proteins. Not only he was able to crystallize this particular enzyme, he also confirmed that this is entirely the protein. So for the first time, the scientific community is able to know that mostly enzymes are proteins. Okay, but this postulate that this particular enzyme is completely protein cannot be applied to other enzymes or other type of molecules. So although he postulated that this particular enzyme is a protein, he postulated that all the enzymes are protein. Then from his findings, he was giving this particular definition that he postulated that all the enzymes are protein, but at that time no one was able to believe this particular definition of enzymes. So what happens then after 1930s or around that time in 1930s, there are other enzymes or the other scientists who were able to crystallize pepsin, trypsin, and other digestive enzymes, and they also found that these enzymes are also proteins. So then they also gave the same idea, and the scientific community, or this idea, which was a controversy for some time, for some 3-4 years. In 1930s, Sumner's conclusion is widely accepted that all enzymes are proteins. Only when certain RNA species were discovered to be having some catalytic functions, then these exceptions will come. Otherwise, most of the enzymes are found to be proteins. During this period, and another scientist called JBS Haldane, so this is JBS Haldane, and he gave, and actually he was writing something. So he wrote a treatise. This is called enzymes. So this enzyme term came from the Pune, and then he started writing something about enzymes. Although the molecular nature of enzyme was not yet fully appreciated, because he was able to crystallize uh, this particular enzyme, urease first, then pepsin, trypsin also, and other digestive enzymes. But we are knowing nothing about at that time about the enzymes. So Haldane made the remarkable suggestion. So this particular scientist, JBS Haldane, give a suggestion that the weak bonding interactions, which are called non-covalent interactions, are required for an enzyme to catalyze a reaction. Okay, so this is very important for enzymatic catalysis, because this type of interactions and this type of suggestion was given by JBS Haldane only. So when he was writing about enzymes, so about the mechanism, firstly, he was able to give this particular point. He noticed that certain weak interactions or non-covalent interactions are happening between the enzyme and the substrate. And today, this insight lies at the heart of our current understanding of enzyme catalysis. Okay, so today we know this is happening, but at that time, this was a big statement. Okay, now we know mostly they are proteins, and there are only certain RNA molecules. Beside this, we should understand certain other concepts. So because we know, and okay, so let me finish this first. So someone has asked a question. So what is the difference between the primary, secondary, tertiary, and quaternary structure of protein? So the primary structure of protein, we know this is the simple sequence of amino acids which are connected by some peptide bonds, or we call amide bonds, CONH bond. Then these primary chains of amino acids will achieve some kind of structure. These structures may be of two types. Either they are alpha helix, if they're forming some helix-like structures, so they're called alpha helix. If they're forming some pleat-like structures, then they are called beta-pleated structures. Now, what is the difference between these two structures? There are various differences, and the most important of all is the hydrogen bonding. Generally, there are differences in hydrogen bonding and bonding pattern in alpha helical and beta pleated structure, and therefore they are given different names because they are forming different kind of structures. Then if they will further fold into a complete 3D structure will be formed, and this 3D structure is having some kind of structure and function relationship. Okay, then there is more than one proteins, then this is called quaternary structure. Okay, but if we talk about the secondary structures of protein, that is alpha and beta, and we compare this alpha beta with hemoglobin alpha beta. So this is completely different. Only the name is same. Here also they are referred to as alpha and beta because of this difference in hydrogen bonding and difference in the type of structure which is formed from this amino acid sequence. But in hemoglobin also there are two types of subunit. One is alpha, one is beta, and this tetramer, two alpha and two beta subunits are forming a tetramer, and it is the 97% of hemoglobin in adult humans. Okay, so okay, then there are certain other type of molecules also, gamma, delta, epsilon, zeta, which are coming under these two classes, either alpha class or beta class, which are present over different chromosomes, either 16 or 11 number of chromosome in humans. So I want to conclude this discussion like this. This secondary structure of protein and the alpha, beta, gamma, delta subunits of hemoglobin are completely different. They are having nothing in common. Okay. So here we are telling because of their structure, they are called like alpha, beta. There they are called alpha, beta because they are encoded from two different chromosomes. So, and they are having something commons. Okay, they are having sequence similarities. Therefore, they are called either alpha globin family or beta globin family. So they are classified like alpha, beta, gamma, delta, epsilon, zeta. So this kind of convention is a kind of numbering system, nomenclature system. Whereas here they are only alpha, beta structure. So hope this is clear. Whoever asked this question, but this is, I think this discussion. Now we are having certain points which need attention in this particular class. These are called cofactors. They are called holoenzymes, apoenzymes, apoproteins. They are prosthetic groups. Okay. So first of all, we will move to another PDF. So here you will be able to understand it in more detail. So the enzyme can be a simple enzyme if there is nothing bound to that particular protein, and the enzyme is pure protein. Okay, so it is made up of only amino acids. So this is called simple enzyme, but mostly enzymes are not simple. They are called conjugated enzymes, or these are called holoenzymes. Holo is complete enzymes. Why this called holoenzymes or complete enzymes or conjugated enzymes? Because mostly the enzymes are interacting with either some cofactors or some non, this is not proteinic part, this is protein. Okay, so some non-protein part of that particular enzyme is interacting with that particular protein is and also helping in the catalysis. Okay, so these are called conjugated enzymes, and if this cofactor and the protein part, so okay, so the two terms will come from here. One is called apoenzyme and one is called cofactor. So cofactor is something which is bound to the enzyme protein part. So this protein part is called apoenzyme, and the non-protein part of enzyme is called cofactor, which is interacting with this particular enzyme to make it holoenzyme, and it will help in the catalysis process, catalysis mechanism also. So cofactor, now we are coming to cofactors. These cofactors can be divided into two major types. Okay, so these cofactors are either loosely bound or they are tightly bound. If they are tightly bound, then they are called prosthetic groups. The prosthetic groups can be of two types, either organic or inorganic. If they are organic, so they are called coenzymes. Now you are able to differentiate the cofactor from the coenzyme. So anything which is bound to protein part and which is having a nature of it is a non-protein part or mostly uh, okay, so, and it is if it is the cofactor the if the cofactor is tightly bound, then it is called as, and it is organic in nature. Second requirement is it should be organic, because there are certain cofactors which are inorganic in nature, or they are metal ions, simply metal ions. So for the coenzyme, the two requirements should be met. So it is a kind of cofactor. Coenzyme is a kind of cofactor which is tightly bound to the protein part, and it is actually the non-protein part and generally having organic nature. So if you give some examples, so the vitamins are the best examples. Okay, so they are organic in nature, they are tightly bound, and okay, so they may be called as coenzymes, or anything which is organic in nature can be called coenzyme. Then there is inorganic prosthetic groups. So they are also tightly bound metal ions. So some of the metalloenzymes you should know. And there is another category in which these are loosely bound. So these same metal ions, metal activated enzymes are loosely bound generally. So the metalloenzymes are kind of enzymes which are having tightly bound enzymes, where tightly bound metal ions with the enzymes, whereas if they are loosely bound, they are not generally metalloenzymes. There, the metal ions are required, but only to activate the enzyme. Then there is no. Okay. So certain vitamins, for example, vitamin B1 is called thiamine. So here are given some vitamins act as coenzymes. So also these all things have been described in your Lehninger also. So beta 1 is called thiamine. Beta 2 is called flavinadenine dinucleotide or FMN. It may be of two type, FAD or FMN, mononucleotide. Okay, so one question has been asked from the FMN portion also. Okay, so how many nucleotides it is having in flavin mononucleotide, because it is written like mononucleotide. So you may get confused here. So this is not single nucleotide. So it is having no nucleotide. The answer is zero in this particular question. If you solved some previous year question papers of CSIR, then you will find this particular question also. So this FMN, which is mononucleotide, is having zero nucleotides. Okay, so this is not part of this particular structure. Okay, then B3, vitamin B3 is called NAD, nicotinamide diadenine dinucleotide, NADP, which is responsible for NADH and NADPH production. B5 is called pantothenic acid. B6 is called pyridoxine or pyridoxal phosphate, that is required for transamination reactions and of amino acids. Amino acid biosynthesis is surrounding around this PLP, pyridoxal phosphate. Okay, so this is vitamin B6 component or the coenzyme. Beta 12, or this is called vitamin B12. This is also called cobalamin, is also is cofactor in some of the enzymes. Coenzyme. Then there's biotin. Biotin is also required some carboxylation reactions. For example, after the first step in Krebs cycle, where the pyruvate is getting converted into the acetyl coenzyme A. So biotin is required there. So this also has been described in Lehninger. So you can find these things from here. So this all we have discussed. So, okay, so the cofactor for enzymes, so you should remember some of the basic metal ions. So, for example, this copper ions are required for cytochrome oxidase functioning. Where you find the cytochrome oxidase? In the mitochondrial inner membrane. So, it is responsible for the electron transport chain. Whereas this iron Fe2 positive or 3 positive is also found in some cytochrome oxidases. Okay, it is also a major enzyme of catalase and peroxidase. So catalase and peroxidase enzymes are having active, so these are called metalloenzymes. So they are having some inorganic ions as cofactors. K positive, this question has been asked from in last year's CSIR. So year I do not remember, but I think 2017 or 18. This K positive is required for pyruvate kinase in glycolysis pathway. Okay, most of the enzyme in glycolytic pathway, for example, hexokinase, glucose 6 phosphate, and pyruvate kinase also, the same enzyme which is requiring this K positive ion, can also catalyze the reaction in presence of magnesium ions. Whereas this manganese ions, Mn2 positive, is required for arginase and ribonucleotide reductase. So, sir has taught you this ribonucleotide reductase and nucleotide biosynthesis. So this ribonucleotide reductase enzyme is having this manganese ions, Mn2 positive. Also the arginase enzyme in the urea cycle, or you have studied somewhere, mostly in urea cycle. It will require Mn2 positive, whereas FEMO, if you know, this is a dinitrogenase complex, nitrogen fixation. So this FEMO protein is also required in this particular complex, dinitrogenase. Whereas nickel is required in urease. So the first enzyme which was crystallized was urease. So you can write a note here. This was the first enzyme to be crystallized and identified to be protein. So by Sumner, okay, so this is requiring nickel ion. Then this is also has been asked. So zinc ion is required by carbonic anhydrase, alcohol dehydrogenase, which we were discussing, and carboxypeptidase A and B. Most important is carbonic anhydrase, because we know in RBCs this carbonic anhydrase is required to convert CO2 into H2CO3 when there is a reaction between CO2 and H2O. Then this H2CO3 is further degraded, further converted by this carbonic anhydrase into H positive and HCO3 negative, so that the it can be it can move from one place, or from one tissue into the lungs, okay, by blood. Now there are certain coenzymes which are the functional groups in certain enzymes. So we were discussing this. Okay, so there are three columns. First is called coenzyme, which is actually the active part in the enzyme, that is called biocytin, for example, or coenzyme A. So certain questions can be asked from this table also. This table is also very important. So you need to learn this table. Okay. So for that, actually, you should know something so that you can understand this table in more better way. So first of all, we need some of the dietary precursors required in mammals. These are called vitamins. So biotin, for example. And one thing, if you have noticed from this particular PDF, that most of the coenzymes are vitamin B complex. Okay, so there are other vitamins also, vitamin A, D, E, K, C also, but mostly in most of the cases vitamin B is required as coenzyme. Okay, so, or the cofactor. So we were discussing this. So if we take biotin in our diet, it will be converted into biocytin. So then this biocytin is acting like the coenzyme in the enzyme reactions. And what type of reactions it will catalyze? It will involving the CO2 transfer. Okay, so the chemical group which are being transferred from one substrate to other, or like this. So the carboxylation reactions, CO2 involving reactions, are catalyzed by these biocytin coenzyme, the enzyme which is having this biocytin. Then there is coenzyme A. This coenzyme A is obtained from pantothenic acid. Okay, and what is the pantothenic acid? Pantothenic acid is vitamin B5. So you need to remember this also. Okay, this B5 is pantothenic acid, and it is a coenzyme A which is required for the transfer of acyl groups. So here, if the acyl group need to transfer from one substrate into the another, you will require coenzyme A. Then there is cobalamin, that is coenzyme vitamin B12. So first of all, the direct question is, what is the active component in this cobalamin, or this particular vitamin? So cobalt is actually present, and it is required for hydrogen atom transfer or alkyl group transfer. So coenzyme A was required for acyl group transfer, whereas this vitamin B12 is required for hydrogen atom or simply the alkyl groups. Alkyl groups are the chains of hydrocarbons. Okay, So alkyl groups, and the active component which we require in diet is vitamin B12. So this B12 is required in diet so that we can do these reactions in our body. Then there is riboflavin. And one thing we should also know, vitamin B and C are water soluble vitamins. If we take them in excess also, the nothing will be harmful. They will be excreted out of the body. Whereas some fat soluble vitamins, for example, A, D, E, and K, are generally fat soluble vitamins, and therefore they are harmful, whether they are present in excess or obviously the dietary deficiency will be harmful. Now there is vitamin B2 that is called riboflavin. Okay, so this flavin adenine dinucleotide is a coenzyme, and we need vitamin B2 or riboflavin in our diet. Why we need this in diet? Because we need many electron transfer reactions and metabolic pathways. To catalyze these electron transfers, we need the vitamin B2. Also, there is an coenzyme that is called lipoic acid or lipoate, that is also a cofactor in the same reaction after glycolysis. When we enter into the Krebs cycle, when this pyruvate is getting converted into the acetyl coenzyme A. So the lipoic acid LA, which is called LA, lipoic acid or lipoate, it is required for electron transfer or acyl group transfer, and most importantly, it is not required in diet. So our body can synthesize this one. Then there is adenine dinucleotide that is called NAD. So this you should remember. This is very, very important. Okay, so this NAD, nicotine adenine dinucleotide or NAD, is required for this hydride ion transfer. Okay, so this H negative ions are transferred by this nicotinamide dinucleotide. And from where we are actually getting this? From the niacin. And what is the niacin? If you do not remember niacin, so let me move you again to this PDF. This niacin is not given this. Okay. This is important actually. This should be there. So because we know most of the coenzymes are vitamin B complexes, so niacin is vitamin B3 actually. Okay, fine. So this B3 is actually the niacin, and it is making one active component that is called coenzyme, that is nicotinamide adenine dinucleotide, and it is required for hydride ion transfer. Hydride ions transfer. So this is vitamin B3. Then there is vitamin B6 that is called pyridoxal phosphate, PLP, or pyridoxine simply. So this is required for very important, and this should because the amino acid biosynthesis is revolve around vitamin B6. So this the transfer of amino groups to make this pyridoxal phosphate, PLP, this pyridoxine is required, that is coming from vitamin B6. Then there is folic acid, and you should know this folic acid is something responsible for absorption of iron also. So this folic acid or folate is making some coenzyme called tetrahydrofolate, THF, and this tetrahydrofolate is responsible for transfer of one carbon group. So if you want to transfer one carbon from one substrate to another, you will require tetrahydrofolate, which is coming from folic acid. And most importantly, what is folic acid? So if you have Pathfinder, page number 100 in 7th edition, about folic acid, it is given in your book. Folic acid is composed of one important ring that is called pterin ring, and which is attached to PAVA, that is para-aminobenzoic acid, and is conjugated to one or more glutamic acid amino acid residues. The active form is called tetrahydrofolate, and the enzyme which is dependent over it, dihydrofolate reductase enzyme, and if there is a deficiency in of the folate in the diet, if you do not take the proper amount, required amount in diet, so it may lead to some disease that is called megaloblastic anemia. So FA will lead to MA. So this is called megaloblastic anemia. What is the meaning of this term? So this is caused due to the diminished synthesis of purines and thymidine actually, which will lead to the RBC, because this is anemia, megaloblastic anemia. So the shape of RBC will change, and they were inflamed RBCs in circulation in blood. Okay, so you should remember this also. The deficiency in diet may lead to a disease that is called megaloblastic anemia. Then there is another coenzyme that is called thymine pyrophosphate. This is vitamin B1, thymine pyrophosphate. So we need thymine in our diet, that is vitamin B1, and it will be converted into a coenzyme that is thymine pyrophosphate, and this is required to transfer the aldehyde groups. So if you search some previous years CSIR papers, so match the column question has been asked from this particular table, in which these coenzymes and these amino acids, sorry, these chemical groups transferred are given, and you need to match the appropriate ones. So if you remember this particular table, this will be easy for you to find the exact matches. Okay, so this we have discussed. Then we are moving to, so in the introduction, these things we have discussed actually. Okay, one more thing actually, what is enzyme commission number? Okay, so this we have left. So this text is also very important, which is so many enzymes have been named by adding the suffix "ase". So suppose there is an enzyme, name an enzyme, and I will tell you it will be followed, or it is having some suffix called "ase". So whenever a term is having this "age" at the last, it mean it is an enzyme. So this is the best way to identify an enzyme. This urease catalyzes hydrolysis of urea, and DNA polymerase, it is also having ase, catalyzes the polymerization of nucleotide to form the DNA. So actually these terms were first given like based upon their function. So whatever the function of that particular enzyme, the name was given in that way only. Okay. So then certain other enzymes were discovered, and their nomenclature has been modified. Other enzymes were named by their discoverers for their broad functions before the specific reaction catalyzed was known. So the enzyme is having certain function, so the discoverer will name that particular enzyme accordingly, whatever the function of that particular enzyme. For example, urea is hydrolysis of urea, and the DNA polymerase is catalyzing the polymerization of nucleotide to form the DNA. Then, for example, and another example is given here. So discoverer find that this particular enzyme is responsible for in the digestion of food, and it is named like pepsin. Pepsin, Greek pepsis means digestion, and then lysoenzyme was discovered. So lysoenzyme is responsible for breakdown of certain bacterial cell walls. So because it is lysing, and because it is degrading the bacterial cell wall, it is lysing the cells. Therefore, it was named like lysozyme. So the enzyme which is responsible for lyse or breakdown. So the discoverers, whatever the function of that particular enzyme, so they named them accordingly. But beyond these native names which were showing their functions actually, they are not followed by some A's actually. The pepsin is not having some A's, or lysozyme is not having some A's. Okay, so they were named according to their functions. But still other were named, there are other enzymes which were named for their source. So firstly, initially when the enzymes were getting their names, so first of all they were named according to their functions, then based upon their sources. For example, trypsin. Trypsin is named because the Greek trypsin means wear down. So was obtained by rubbing the pancreatic tissue with glycerin. If you rub the pancreatic tissue with some tissue glycerin, so it will release some of the enzymes that is called trypsin, which will help in the degradation of some meat or something, some food. Sometimes the same enzyme has two or more names also, because in different parts of the world, the enzymes were discovered and the scientists are giving them name uh, either conventionally based on their source, based on their function, and so on. So it is also possible that same enzyme may have two or more names. Okay, or it is also possible that the two enzymes is having the same name. Okay, so two different scientists in two different parts of the world discovered the same the enzyme, two different enzymes, and gave them the same name. It is also can happen. But उसके बाद क्या हुआ actually, all the biochemists or all the scientists who were doing the biochemical or the protein research or enzyme research, then they make a committee. That the committee is called as International Union of Biochemists and Molecular Biologists. So this nomenclature committee of international scientists from the two fields, either from biochemistry or molecular biology, in 1956 agreed that they will name every discovered enzyme according to the rules which were designed by this particular committee. So in 1956, the committee will came. Okay, so because there are so much ambiguities in this classification and nomenclature, as well as there is ever increasing number of newly discovered enzymes, because so many enzymes were discovered in the different parts of the world. So these biochemists by international agreement have adopted a system for naming and classifying the enzymes, and this system divided enzyme into the 6 classes with subclasses based on the type of reaction they catalyze. So the subclasses, if someone asks you this question, so these subclasses are formed based on the type of reaction, or these 6 classification system was based upon type of reaction it was catalyzing. Each enzyme is assigned a 4-part classification name and a systemic name actually, which identifies the reaction it catalyzes. As an example, we will discuss 2.7.1.1. So it is given there, or we will discuss. Okay, move. So for example, this is given. Okay, so for example, the first enzyme in your glycolytic pathway, that is called systemic name is ATP dehexose 6-phosphotransferase. मतलब ATP से phosphate group को hexose is any glucose or anything means which is having 6 carbon. So this is called hexokinase in trivial nomenclature, but when it is given a specific name, so the donor is written like ATP column, the D-hexose is the acceptor, and 6-phosphotransferase at the 6th position of this hexose. So this is transferring the phosphate group. Okay, so this is called kinase actually. And what is the EC number? So this is called enzyme commission. So the enzyme commission is giving a 4-part name or a number to each enzyme. For example, this is called 2.7.1.1. So what is meaning of this number actually? So I will discuss this also. The first name denotes the class name actually. So let me show you what enzyme commission has given some rules. according to Enzyme Commission, the 6 classes of enzymes were designated as 1, 2, 3, 4, 5, 6. So one mean oxidoreductases. So those enzymes which are doing oxidation reduction reactions. The second type of symbolizes the transferases. So the enzymes which are involved in group transfer reactions. So all the kinases which are responsible for phosphate group transfer are coming under this particular class. Third is hydrolases. So hydrolysis reaction or transfer of a group to the functional group or to the water molecule is called hydrolases. So these group involve the hydrolysis of water, whereas lyases, isomerases, and ligases similarly. So actually we know the second is denoting the transferases, and we know the kinases are also transferases because they are transferring the phosphate groups. So if someone asks you the kinases, so these are all transferases. So therefore the first number is 2, because the first number 2 denote the class name that is called transferase. Second number 7 is a subclass, and this is called phosphotransferases because they are transferring the phosphate groups. Third number is 1, so this number is a subclass. So actually this is showing the phosphotransferases with the hydroxyl group as acceptor. So this OH group is accepting this phosphate group first. Okay, and the fourth number 1 is actually a serial number, which is denoting that the D-hexose or D-glucose is actually the phosphoryl group acceptor. So if the glucose is acceptor, then it is 1. If something mannose or some other hexose is acceptor, then this number will change. So that's how the 4 number either divided by points is showing a specific enzyme with a specific function. So this is more systemic way to classify the enzymes. Okay. So all these things we have discussed, and this is written here, nomenclature committee, that is the International Union of Biochemistry and Molecular Biology. It was formed in 1956. You can find the link even here. Okay, so they devoted primarily to the principles and properties which are common to all of the enzymes. Now, what we have discussed in the introduction part of the enzymes, we have discussed that life depend upon the 2 things. One is self-replication, but the second most important is the powerful and specific catalyst, which are called enzymes, so that the biochemical reactions or the metabolic reactions can take place. With the exception of a few catalytic RNAs, all known enzymes are proteins. Okay, so they are having either coenzymes or cofactor which are required for the catalytic function, and these can be either loosely bound or tightly bound. Okay, so mostly the coenzymes are tightly bound. Then there is enzymes are classified according to this enzyme commission numbers. Enzyme commission. Okay, so the numbers are given in accordance to some of the rules which are designed in this particular table. Okay, so we will start our discussion in our tomorrow's class from this particular table, how the different enzymes are classified according to this EC numbers. Okay, so further we will discuss